So welcome to G05 Antibody Analysis Demonstration Video 1. We're looking at immunoglobulin sequence annotation. So we know that the regions at each ID domain are very, very diverse. And then we have different domains, the FR1234 and CDR123 domains that we want to annotate. The CDR regions are especially diverse. And we know that annotation tools are centered on human and mice and other related well-studied organisms, less so for rarer livestock species where they may have different antibody structures, such as the long CDR3 region in certain Bos torus samples. So what we're going to use is a tool called IgMAT, which is based on the ANARCI tool, which uses data primacy from the IgMT numbering scheme. And this uses existing known antibody and TCR alleles, as well as a curated antibody germline set as well. So IgMAT stands for immunoglobulin multi-species annotation tool. It uses hidden mark models to look at this sort of data. And what we're going to do then is look at that in more detail in our piece here. So just be warned that IgMAT may be limited by short sequences, are also the input that if we've a very novel antibody, it may struggle with annotating that correctly. So what we're going to do is access that website on GitHub as outlined here. And then we're going to go to that folder, create a Python environment, activate that environment, finalize the installation, and then we're going to run queries like the example one given here, where we can use the run function given some sequence input, and verbosely give us the output, and we'll get something like this, where we see sequence alignment of this test sequence and its annotation for the FR1, the CDR1, FR2, CDR2, FR3, CDR3, and FR4 regions as outlined there. We can also provide this as an input FASTA file instead of having a sequence like that. So we can create a file test.fa with any FASTA file, any immunoglobulin files of interest. And what we can do is add on that a file name extension for that folder, AN, for that folder, and we'll create a folder called new one, where we can open up firstly a bed file, then the output, which we'll see like that, as well as the alignment itself. So we see there the FR1 region spanning from amino acids 1 to 25, and so on. So for example, if we take this pig BDJ region, we'll see an alignment like this on top, and then we see an output like this containing the regions that are of interest. What we'll do as well is run this on a larger number of Bos torus sequences, 65 in total, and we'll see how that works. So we're going to switch over to the server now. Okay, so what we're going to do is copy over that GitHub website for IgMAT that we want, and then we're going to go into that directory. The next thing we're going to do is set up the Python environment, and then we see that's set up now because we have the env symbol there at the end. We then need to activate that environment. So you can see the env is now set up properly. And then we need to do pip install to install this tool properly. Just we / to indicate this directory, and that will install IgMAT at this directory properly. We can next test that installation using the IgMAT run command. And here we're going to give it just a simple input sequence, which you can see there, that amino acid sequence, and we use the tag -b to indicate we want verbose. Our output, if we don't include -v, we get nothing. So that's not useful. What we can do is copy over a test query. I'm going to copy test.fa here, and we're going to modify our command. So IgMAT run -input is test.fa, and we're going to call it -an annotation, as we're going to call it new one. We want verbose output as well. So there we see our alignment for this test.fa, which, as you can see, is the same or it's different sequence to above. We can see all the regions are identified. And if we look at the output for new one, we see a set of files. So first you have the FASTA file. Next we have the bed file. Look at the bed file just to notice the locations where the FR1, CDR1, etc. are all present. And then we have the alignment file containing that alignment as shown above. So that's quite useful. And then what we can do as well is batch commands, batch queries. So if we copy a file containing a bunch of boss tours, VDJ sequences here, what you can see is that they're contain IG heavy chain regions. There's 65 of them here. So again, we run igmat-run-input bos.fa -am, we're going to call it bos1 -verbose. And we see all the alignments being produced there. And now we can look at bos1.bed for each of the coordinates for each of the samples there in sequential formats. So you can see the first one there is the first 7 lines and so on. And we can see the same for the alignments. We can look through them all as well too there as we go along. So we'll talk about this more in the lab, and you can query your own sequences that are of interest using the same approach as well. So please bring on your own questions as well.